Our next session involves COVID treatments and other things. And our first speaker is James McMahon, who's an ID physician at the Alfred and Monash. And James is going to talk to us about COVID-19 treatments. Thanks, James.
No worries. Thanks, Lou. So yes, ID physician declarations. I am on the COVID evidence task force that Bridget chairs on the treatment panel and lead a current MRFF clinical trial of COVID vaccine boosting in immunocompromised hosts. So that's probably the main things. I also have many roles with Asham, and I notice Alexis is here too. And Alan is now my new boss, so I've got to say the right things about Monash Health. Just trying to go forward. So we're going to talk, this is a bit of a work, so I'm going to try and focus on evidence and trials and let Bridget talk about task force and procedures more. Essentially there's two big domains, early disease and hospitalised disease, and we've got more and more options in that late category, which I'll move through more quickly. So I've just had this one cartoon which I constantly modify to show the natural course of COVID-19 and then how the recommendations of the task force have changed, and that's currently, I think, fairly accurate. Bridget obviously will correct me if I'm wrong. There's many conditionally recommended drugs in early disease and in established hospital disease that requires oxygen there. I'm going to start early disease. So we have two main drugs that have had a lot of interest. I still like this cartoon, which is from a long time ago in science. So we have nirmatrelvir ritonavir boosted with the P450 inhibitor ritonavir as a protease inhibitor of SARS-CoV-2. And then we have the Merck drug molnupiravir, which acts on the RNA-dependent RNA polymerase to inhibit RNA replication. And I'll have a little chat about these two medications. So essentially, this is the main study that was in the New England Journal of Medicine that led to the licensure and widespread use of this medication to prevent the progression of SARS-CoV-2 infection to severe disease. The most important things about these trials is they are done in unvaccinated individuals, and they're done in an era well before Omicron. So they're very different clinical studies, and in fact all of these studies were intentionally done in these unvaccinated populations to make sure we get a clean signal. They had a risk factor for severe disease which was often fairly mild, and it might just be age or it might be elevated BMI above 25. This was a standard treatment course of 5 days of nirmatrelvir ritonavir, and you can see there the very large delta. I cannot read that well on that slide, so I'm going to cheat. And the efficacy, oh, thank you, someone's just done something really clever there on my screen. To see that risk benefits between Paxlovid or ritonavir and placebo, 80% odd benefit in preventing progression to its primary endpoint of hospitalisation or death. The main thing with clinically with nirmatrelvir ritonavir that we're constantly dealing with is the issue that ritonavir and the interactions you can see that are listed on the insert. I've just highlighted some of the more common drugs that might be prescribed. Essentially, there's a wonderful website, COVID-19druginteractions.org, and the HIV clinicians are very used to doing this sort of process and understanding it, and we must encourage our clinicians to constantly check for these drug interactions and still try and use this medication. So that's the story with that medication is being used more widely. Molnupiravir, named after Thor's hammer, Mjolnir, hence its name. Again, similar study, 5 days of medication. These are unvaccinated individuals with symptomatic disease looking at the progression to hospitalisation. And this was this staged releasing of results where the first interim analysis that was released had that 50% efficacy in prevention of that progression versus the final data where the subsequent population really didn't get as much effect. So the ultimate efficacy of that prevention of progression of hospitalisation was about 30%, and it can't be used in pregnancy. So that's the NEDGEM study, led to its licensure, and this again is the Kaplan-Meier plots, which shows this moderate benefit in preventing hospitalisation, and on the right-hand side you can see that the subgroup analyses, there's really no difference between multiple different factors, the severity of the disease, their gender, etc., in that modest benefit. So we have that data. And then I'm going to spend a little bit of time talking about this, and I've limited this talk to discussing the randomised trials in general. So this is the panoramic trial from the UK. So, as I mentioned, these original registration studies for these agents were done in unvaccinated population, which is not the reality, or also previously uninfected people. And what we've always wanted is large randomised trials in vaccinated people, and that's what this is. This is a very large study done in the United Kingdom, and they've published on molnupiravir recently, and they are also studying nirmatrelvir ritonavir. This is an open-label study, and it started in late 2021. You had to be over 50, or you could be younger than that with some comorbidities. And again, you were randomised to molnupiravir twice a day for 5 days, or usual care, so that you knew what you were on. High-risk people could get other drugs, such as monoclonal antibodies, which were being used at the time, and that was recorded, although in the published paper it was very rarely done. It was done with online diary for 28 days, and people had to report their symptoms as having no problem, mild problem, moderate or major problem, how they were feeling. And this, again, is open label. People know that they're on the drug or they're not, but the primary outcome was hospitalisation or death, as it was done in those previous industry registration studies. This was assuming, assuming that 3% of individuals would progress to this outcome, and they were powering it to reduce that by a third to get the outcome of that progression from 3 to 2%. So they thought they did 5,500 people in. However, they found that their event rate was a lot lower, so they changed their sample size to 16,000 people for 90% power, or 12,000 for 80% power, to get their event rate, which is what was occurring. 1% of their patients were progressing, to get that down to 0.2%. And what they ended up doing was they ended up stopping at that 12,500 number, that 80% power number. So the primary outcome I mentioned was this progression, but that was actually done in a regression model. And there were factors that were controlled for, as you can see there, age and vaccination status and comorbidity. There was meant to be a range of interim analyses as this went along to sort of try and find the answer, as adaptive trials do. But actually what happened was there was no interim analyses, and the first interim analysis was done at 12,500 people, which seems like a very long time to wait for an interim analysis, but it's because they enrolled quite quickly. This may not show up as well, but essentially what I'm trying to show you there is this is an enormous amount of people. 100,000 people were potentially screened. 68,000 were put forward for the eligibility check, and 30,000 were considered ineligible to get to this bottom number of 26,000 people were randomised within 12,000 people in these arms at the bottom. So an enormous randomised trial done in a vaccinated population. And that really probably, if you want to look at those tables, the baseline, probably the most important one is the one I've boxed there, that as you can see, 92% to 3% had 3 doses of vaccine, and essentially no individuals had received no vaccine. And a small, some people had inhaled corticosteroids, and less than 1% of individuals had a monoclonal antibody. So it's a highly vaccinated population in the UK, and they hadn't really received many other treatments that we're aware of. So essentially there was no difference, is the short story on the primary outcome, between hospitalisation of death, 1% basically in each arm, and that there was a difference in the time to recovery of 9 versus 15 days, and the median time to first recovery was 10 versus 15 days. So these met the threshold for superiority, but as mentioned, it is an open label study, and it is the patients being asked, do you think you have recovered? And there is many examples in the literature of that potentially being a softer outcome when you know whether you have received the drug or not. There was a virology sub-study, and there was, again, some evidence of a mild virologic effect, but it was very small numbers of individuals in that sub-study, only 75 odd people. This is the Kaplan-Meier plots. On the left is the subgroup analysis for hospitalisation or death primary outcome, and really that's just to say that you can look in the subgroups, there's no major differences by those different groups. and on the right is that benefit in first reported recovery, again with that caveat that it is an open label study. So you've got these two, this is obviously, the reason I'm spending the most time on this, has been a lot of discussion about this study in the context of the initial registration study from Merck, and they are very different, but they have led to many different interpretations in guidelines. The main thing really being that this panoramic trial was done in a highly vaccinated population during the Omicron wave in the UK. Oh, I've spoke a typo of discussion there, I'm sorry. And I've just really, I don't want it to be too controversial, I've just literally pulled these conclusions from the discussion of the paper. This really, the authors of this trial thought that avoiding this outcome is really done by vaccination and is not done by drugs such as molnupiravir. But it had this very wordy, take a bet every single way sentence as well. In time to recovery, resource utilisation, potentially a virologic effect, healthcare services, drug costs, maybe there is some ways that different jurisdictions may find that there is some benefit into using this drug. Okay, so that's the more longer discussion. I know there's been an observational analysis which is in for publication here, which again I haven't gone through in great detail for time. Happy to answer questions on that though if people want to talk about that. And then the last drug for early disease is remdesivir, and this drug again showed quite high levels of efficacy. Same thing as the previous registration. These are unvaccinated people, have not had prior infection, and they get the drug very early in the course of disease, and they get it's an intravenous medication. And then when you give 3 days of remdesivir to someone early disease, less than 5 days, you get a similar benefit to nirmatrelvir or ritonavir, about a 90% risk reduction in the progression of hospitalisation and death. So that's also an important outcome of the antiviral benefit in that group. I used to have a whole section of monoclonal antibodies, and what I used to do is I'd have big font and little font, and when the drugs got sort of blown away by variants, they become little font. Everything's in little font now. We don't have monoclonal antibodies that we can use, is the short story, and I like this term, and this is Stuart Turville's paper, and what I like about preprints is that you can have a preprint, and that things can change so fast in COVID, you've got to update the preprint. So the preprint goes to V2 and V3 and whatever. And this is, I mean, Tony's here, can sure talk to this, but like this is V2. I got this off the web recently, but this has already moved on. And if you can understand this, you're a smarter man than me. But it is fascinating just looking at, you know, we've got Deltacron down here, and we've got the BA2 ones here, and the BA5 ones down here, and XBP recombinant from the 275 to the 1.1, and if you get on Twitter you can get completely confused as all the sort of the virologists start chatting about variants and all this sort of stuff. But I think the variant soup is a great way of describing it, and that's what we exist in now. And this is again some of the work from Stuart Turville and Tony, showing that basically these different antibodies, the one we've sort of most been interested in is Evusheld, has really lost activity against the variants that are currently circulating. So we're not using that for prevention or treatment. And I think these are great. I love this CDC website. It's amazing how the CDC puts this variant data online every week in real time, and you can just watch the evolution of these variants. And we've got all these different things circulating at once. In Victoria, we do rely, I think I'm correct in saying that the Chief Health Officer media release, there is a paragraph in there which you can get into and you can sort of work out what's going on in Victoria, but we are dealing with similar lineages to the US, and XBB 1.5 is the one that's the most exciting at the moment, but we don't have a lot of that as of 30 January, but we're a lifetime ahead than that now. Already. So watch this space to understand how these variants will go with time. So we have these sort of, again, these tables with the ideas of how efficacious these drugs are, and essentially we've got one study here in a really highly vaccinated population, and they've got these 3 other sort of industry studies showing this 85, 30 and 87% risk reduction in these unvaccinated individuals. And you can see that the event rate was low in the panoramic trial, and the people progressing is similar to the progression rates in the people who got the drugs in the industry studies, whereas the progression rates were higher in the unvaccinated people that got placebo in those original studies. But this is that's the data to support these drugs. So really, this is nirmatrelvir ritonavir is first line, remdesivir is another option, and panoramic really doesn't support the routine use of molnupiravir, and antibodies are out. Okay, so I've got 5 minutes left, and I'm going to try and fly through the drugs that we now use in the hospitalised setting, which is expanded, and I'll just keep it punchy. So basically, a lot of this is really driven by the recovery trial, which I'm sure we all know. And this was, I looked this up, this was June 2020, which is amazing that was when the media release was about dexamethasone use. It's sort of getting up towards 3 years. And really to show this mortality benefit up to 30-40% in the ICU setting for 6 milligrams of dexamethasone routine care. And all the other studies that have looked at corticosteroids in the meta-analysis have showed this similar benefit to using corticosteroids to prevent death in hospitalised COVID-19. The high-dose paper was released on preprint in December, so there's still actively people looking at this question. There was a big debate about higher doses of steroids, would they add more benefit? And the answer is, if you're just on face mask oxygen or intranasal oxygen, it actually is, you are worse off. Somehow they hit the sweet spot with 6 milligrams of dexamethasone the first time they did it, and these higher dose of 20 of dex followed by 10 of dex worse outcomes, and they also increase pneumonias. They're not COVID, they increase insulin requirement and things like that. So that has not been published as that I'm aware of, but it's in preprint. Remdesivir, we use this in the hospitalised setting, and the font's gone a bit funny there. This was the study that Fauci released in the White House with Donald Trump way back at the start of the pandemic, showing that this drug shortened time to recovery in an unvaccinated population in hospital. And then subsequently there was a large study done across countries all through Europe and the Americas and Asia, which showed that this drug, it's the top left panel there, did not have a survival benefit. So we had this conflict between this large NIH-funded study that showed this benefit in recovery, placebo control, versus a large pragmatic trial which showed that it didn't improve survival. And the short story with that is that these different studies were interpreted differently by different groups around the world. In Australia and many high-income countries, it is still used in the hospitalised setting for people requiring oxygen from some of those early findings. And there's other observational data which I haven't got time to present which support its use in hospital settings. So we've got steroids for survival. We've got remdesivir to assist people early on. And then we've got baricitinib. So baricitinib is a drug used in rheumatoid arthritis and inflammatory bowel disease, which inhibits this JAK-STAT pathway, which is a pro-inflammatory pathway. The cytokine triggers that whole response. Baricitinib blocks that and dampens down the inflammatory state. And the very first study, again, was this NIH group. They were looking at the addition of baricitinib, and again, they had this time to recovery endpoint, and when that was added to remdesivir, that improved that time to recovery, particularly people requiring non-invasive or high-flow oxygen. I was recollecting that each one of these slides used to be about 7 or 8 slides, and now the study is just diminished to this one little slide every time. And then cough barrier came out, and this was probably the most landmark study in this space because they showed a mortality benefit. Again, a large study done, a placebo-controlled trial, and they showed this mortality benefit when you added baricitinib to remdesivir. So that is 80% of people receive steroids in this study. So we do routinely, many guidelines recommend the addition of baricitinib to steroids to limit mortality. I won't go through the Kaplan-Meiers in great detail. And the other important bit about cough barrier, they did another study in the really hard to study population in the intensive care unit, intubated on ECMO, and they showed this impressive benefit in survival by using baricitinib in that setting. So, and this was reinforced by the recovery trial, again, had the most patients, again showing a survival benefit on top of the use of steroids. So we've got steroids, we've got remdesivir, we've got baricitinib, and then we've got tocilizumab, again, an IL-6 inhibitor. So you can get the sense here, we've got this increasing suite of potent anti-inflammatory medications that we can use for people who need oxygen in the hospital setting, and again showed a survival benefit, again on top of the use of steroids. This is a drug that's given as an intravenous dose with a long half-life and possibly a second dose. And again, recovery was critical in understanding tocilizumab because there was actually a huge range of studies that showed variable findings before recovery that were much smaller, but recovery really sealed the deal for tocilizumab that it does have an indication in this setting. And last but not least, I will mention these two drugs which are in preprint and have both shown a benefit. This is a large US government, NIH-sponsored immunomodulatory investigator collaboration called Active-1. And these are basically people hospitalised with moderate and severe COVID-19. And they are looking at whether these drugs, in addition to steroids and remdesivir, provide benefit. And the short story is that they do. Again, it is in preprint. For this benefit of time, I won't go through the mechanism of action of these drugs. These are used in conditions like inflammatory bowel disease and rheumatoid arthritis. But you can see there's a mortality benefit there of about 30 to 38% for abatacept. And then for infliximab, again, a mortality benefit there of about 40% as well, on top of steroids and remdesivir. And these drugs have made their way into some guidelines already. And we end up with things like this, which I'll let Bridget discuss. So now this is the sort of the picture we have for if we had someone in hospital on oxygen who's got inflammatory disease, we've got all these drugs we can use. So I will stop there. Thank you very much.
James, thank you. Terrific. Questions, please. Bill at the back.
So Jamie, two questions. So we're no longer using Eversheld?
Correct. It's not available. The last people would have received it for prevention in about November, early December in Australia.
And in terms of molnupiravir, I mean, clinically there's not a lot of enthusiasm for it.
Yep.
And I guess the other issue is that there's a paper kind of the last few months looking at wastewater and showing promulgation of molnupiravir mutations in viruses going forward. So rather than causing air catastrophe and virus death, cause a mutation, but those mutant viruses survive. So it's actually driving mutations which are potentially could increase fitness, and so there's actually a lot of concern about whether we should use it at all or just stop using it completely so that you don't make more mutant viruses which are potentially more fit and dangerous. Do you have a comment?
I don't have a strong comment on that, to be honest. I think there is some debate both ways. I don't know the paper you're talking about. I certainly know in Victoria there's been a big discussion, and also nationally now, about the observational data in Australia that supports the use of molnupiravir versus data such as Panoramic. It's a randomised trial setting of a vaccinated population that doesn't support its use, and there has been differing interpretation of that in Australia, and there is and actually, interestingly, the editorial, if you're interested, for the Panoramic trial is written by Australian authors, and I understand that there's an observational data set in the conversation, and that's been submitted for publication as well, trying to whether there is an additional benefit. But I just think that I certainly know in the task force process we've not been involved in the using of observational data due to the intrinsic biases and so confounding.
But why not just use Paxlovid?
Correct. I mean, I'm very supportive, and I think that obviously molnupiravir doesn't require the drug interaction issue, so it is easier to implement. But if you're my comment would be, if you're implementing a medication that's potentially not effective because you won't go down the process of looking for the drug interactions, then you're potentially denying someone a drug that could benefit if you just went through that process. And it is a bit of extra effort, to determine if these drugs interact or not and what to do with that. So, yeah.
Yes.
Just to comment on that, the GPs have often prescribed molnupiravir because of the hassle, and even though a number of them have been made aware of the University of Liverpool checker and that people can go through that. More importantly, with the onset of Omicron, what good evidence do we have that the effectiveness of Paxlovid against these more recent Omicron strains in terms of reducing hospitalisation and deaths?
Yeah, we don't have it. There's good data supporting the ongoing efficacy of the directly acting antivirals against variants, in vitro settings. But for me, the bigger question has always been, what is the efficacy of these drugs on top of vaccine? I mean, vaccines, we know, prevent hospitalisation, death, yeah? So Panoramic's doing nirmatrelvir ritonavir. So that same group is doing a large randomised controlled trial of Paxlovid. So that's a really important study to watch those results for. And it, as the conclusions here were, it supports the importance of vaccination. Now, molnupiravir was not as potent as Paxlovid in the registration study, so maybe there will be some benefit of Paxlovid in that panoramic trial. We just don't know. We need to wait and see what it shows.
Rod, we've got 13 seconds.
If they're not dying from COVID now because of your magic, what are they dying of? What pre-existing morbidities do you think are the most at risk?
Broadly, age is the biggest predictor, yeah, and then other severe states of immunocompromised.
Yep.
So, like flag, we're doing the trial in immunocompromised states with vaccines because we think that's important. Yep.
Particularly the transplant group.
Oh, transplant, absolutely, and some of the hematological malignancy groups, absolutely. And rituximab and these sorts of things that they just don't have responses. Yeah.
James, thank you.
Right,
Terrific.
thanks, Luke.